Hi, everyone, and welcome to the Human Cell Atlas Biological Network seminar featuring the oral, craniofacial, and skin networks. So I hope many of you joined us for last week's general meeting. If you didn't, recordings are posted on the event platform website, and one of our staff members will include that link shortly. So the goals of this seminar series are to increase the biological network visibility and to spark new opportunities and collaboration and engagement. It's to promote the 18 biological networks and also to engage the CZI-seed networks. This seminar takes place the second Thursday of each month from 10:30 to 12 p.m. Eastern. And we are very grateful to the Chan Zuckerberg Initiative for making this seminar series possible. So we ask that you submit questions in the Zoom Q&A, which you should see. You can also vote for questions using the thumbs up and thumbs down as well. The seminar format will be as follows: 15-minute talk by Paul Sharp. We'll have 5 minutes for Q&A, and then a 15-minute talk by Magnus Lynch, followed by 5 minutes of Q&A. We will then enter separate breakout session rooms for 45 minutes. And please note that you will not return to the webinar room, which is this room, after the breakout sessions. So the breakout sessions will end, and that will be the end of today's seminar. So I'd like to turn it over to my colleague, Ellen Todris, who will introduce our first speaker. Ellen.
Yes, hello everyone. We're very grateful to welcome Paul Sharp today. Paul Sharp is the Dickinson Professor of Craniofacial Biology at King's College London. He graduated with a degree in biology from York University and PhD in biochemistry from Sheffield University. Following postdocs in Sheffield, Wisconsin, and Cambridge, he became lecturer in molecular embryology at the University of Manchester in 1987, where he established a research group working on the molecular control of tooth development. In 1991, he was recruited to present chair at the Dental Institute of Guy's Hospital, later to merge with King's College, where he established a new basic research department, the Department of Craniofacial Development and Stem Cell Biology. The department, of which he remains head, now consists of 15 academic research groups with over 80 research staff. And in 2017, it was awarded a Center of Excellence status, Center for Craniofacial and Regenerative Biology. From 2002 to 2008, he was Director of Research for the Dental Institute. Paul's main research interests are the molecular control of tooth development, dental stem cell biology, and tooth bioengineering. His current focus is on understanding dental cell function and the development of stem cell-based approaches for new therapies in clinical dentistry. Paul, welcome and thank you.
Thank you, Ellen, very much. Let's just make sure this is going to work. Desktop 2. Okay, can everybody see that?
Yes, it's good.
Okay, brilliant. Okay, well, thanks very much for the invitation. It's a great pleasure to be able to speak to you today. I thought I'd start by explaining my title a little bit. So the way I try to get my lab to view transcriptomics is that it's the latest in a long line of approaches to study gene expression, the modern day equivalent of northern blots, if you like. And that makes it a tool, albeit a very powerful tool. But of course, it's what you do with the massive amounts of data you get that's important. And assuming you started out using transcriptomics to answer a particular question or questions, you will almost certainly answer these, but the data sets will inevitably throw up all sorts of new questions, oddities, and even results that don't make any sense. And I think everybody at some point, therefore, is likely to find themselves cherry picking, looking at interesting observations and following these up. And that's the basis of my title. And I'm just going to give you one brief example of of that today in relation to telocytes and macrophages. But first, to put my lab's interest in transcriptomics into context, hold on a minute, the slides aren't moving. Okay, there we are. So here on the left just shows a pictorial summary of the work that we've been doing for quite a long time now on the mouse incisor. We use the mouse incisor because it's continuously growing, so we have very clearly defined spatial stem cell compartments. And we've been using transcriptomics and other techniques to study those. On the right panel here shows the work of Anna Catino in my lab on using human gingiva, periodontal tissue to study those tissues, and also how cells change during disease, gingivitis and periodontitis. And on the bottom here is work from a student in my lab, where we try and recapitulate some of the human results in mouse models, so mouse model in particular of periodontal disease. And I'll tell you a little bit about this today. But we started the incisor work with a specific question, and that was to try and understand something about the stem cell compartments and how cells transition along this, spatial and temporal axis. So in yellow here, you have the slow cycling stem cells in a clearly defined niche. They give rise to transit amplifying cells, and those transit amplifying cells differentiate into the cell types, and these arrows and molecules show a little bit of what's happening. But we became interested in these blue cells here, which are present in extremely low numbers, and they express a number of molecules, in particular SELSA1. These cells are completely quiescent. Unlike the stem cells which cycle, these cells do not cycle at all. So we became interested in trying to understand what is the function of these cells, and I'm going to cut a long story short, but they act as reservoir cells. So when you challenge the growth of the incisor, usually simply by cutting it, the incisor accelerates its growth. And it does that by mobilizing these quiescent cells, which start to enter the cell cycle, and they give rise to new stem cells. And the transcriptomics shows that very nicely, even without any other data. So here, population 17 are the quiescent reservoir cells, the SELSA1 positive cells. This is just in a normal situation. During regular growth, they really don't do anything. They don't interact with any other cell type at all. If you cut the incisor, within 15 minutes, these cells start to become activated, they enter the cell cycle, and they start to give rise to another population, which then eventually become transit amplifying cells. And that is the way that the incisor is able to regulate its growth at times of need. The transcriptomics also starting to throw up the mechanism of how that happens. So these are the quiescent cells, and SELSA and CDKN1A negative. Within a few minutes of cutting the incisor, they start to express CDKN1A, at the same time still express SELSA, and then they lose SELSA expression and fully enter the cell cycle and become stem cells, basically. So that's the question we set out to address, and we're still working on that. In particular, how does the action of cutting a tooth send a message all the way down to the other end of the tooth to activate these cells? But when we got these data sets, a new student that joined the lab noticed this population here. This is a CD34 positive cells, and very quickly identified these as telesites, often called interstitial cells. And they're a very unusual cell type about which very little is understood in lots of tissues. There are a few things listed here, regulators of stem cell niche, etc., etc. But we thought that these were such odd cells that we would start to investigate them. So the student cherry-picked those populations. And I should say in the incisor, there are quite a lot of these cells. These are what they look like. They look like aliens, basically, something from outer space. They've got these amazing projections that they emit. And it's known that these projections make contact with other cell types and do something. What they do isn't really known in most tissues. There is some information, but really they're very enigmatic cells. And so Guy, who started this work, decided to look at these telocytes in the mouse incisor, and I'm not going to show you lots of data, but one of the things that's really interesting is that these telocytes clearly interact with odontoblasts, the dentine producing cells. And this is one picture here of a telocyte starting to burrow its way into the dentine layer. You can image this in 3D. So we're obviously interested in trying to discover what these cells are doing in the incisor, whether they're necessary for any of what we see in the incisor growth. But I'm not going to talk about that. I'm going to talk about telocytes in periodontal disease, because another student in the lab, Jing, who'd actually produced this image here, decided she wanted to deviate from her main PhD project and also look at these cells. And in particular, ask the question, do they have any role in disease? In this case, periodontal disease. And this is still in the mouse at this point. So this is the UMAP plots here. We compared homeostasis with disease, and the telocytes are just here. When you look at where the telocytes are located by their expression of CD31, they are predominantly seen in close association with blood vessels, but not exclusively. This just shows that a bit clearer, just looking at the telocyte population. So these are clearly telocytes. They have a very close association with these cells here that express many of the same genes as telocytes, but significantly these don't express CD34, which is the sort of archetypal gene that telocytes express. So when we look at telocytes in the resting homeostatic periodontal ligament in the mouse, they are extremely small in number. There's very, very few of them, and we can't detect any evidence of proliferation using all the standard techniques. And we also have an inducible Cree mouse. So if we label these cells at a particular time and chase them out, we just don't see any increase in number at all. However, in our induced periodontitis model, which is a ligature model, which we find the most reliable, and we able to measure the periodontitis induced by bone loss, within 2 hours of releasing the ligature and sacrificing these cells, we see telocytes that we never normally saw any before. There's a big increase. And by 7 days, now using a lineage trace, going from hardly any cells, we see a big increase. You see these purple cells here. So the induction of periodontitis using this model stimulates the proliferation of these normally quiescent telocytes within the periodontal ligament. And we've done a Ki67 to see that, and this just quantifies all that. When we compare the transcriptomics of these cells pre-periodontal disease and subsequently, the top hits are angiogenesis, leukocyte migration, and inflammatory response. So periodontal disease is an inflammatory disease, of course. So it sort of suggested that these cells have got some role or responding in some way to the induction of disease, and hence are interesting. So we did all the usual stuff that you might expect, interrogating the data, and I'm just going to show you one thing here. Essentially, we were looking to try and identify if there is any significant difference between these proliferating telocytes in disease and the resting telocytes in homeostasis. And I'm only going to talk about one interesting molecule, which is HCA, hepatocyte growth factor here, which is not expressed in resting telocytes. Its expression is induced during periodontal disease when the telocytes become mobilized. And in that situation, telocytes appear to be the only cells that start to express HCA. So there has become a unique single source of this molecule, and all this stuff you see here shows that. When we started to look at potential interactions between these telocytes expressing HGF and obviously other molecules and other cell types, the obvious thing that became immediately obvious was potential interactions with macrophages. Now, the macrophages were interesting because in disease, we'd found that there were clearly 4 distinct populations. Others have reported this in other tissues. And the telocytes, at least by this data analysis in silico, appeared to be potentially interacting with at least 2 of these different populations. And indeed, when you look in the tissue, in the diseased tissue, you can begin to see in yellow an interaction between these telocytes producing their projections. Whoops, just jumped ahead. And interacting with macrophages. And this just shows this a little bit clearer in terms of. So telocytes, when you induce periodontal disease, start to divide, increasing number and express hepatocyte growth factor. And the silico data suggests that if they're interacting with anything, they're interacting with subpopulations of macrophages. There's a lot of things on this slide, and I'm just going to, in the interest of time, go through them very quickly. So we then reclustered the macrophages, these 4 populations, just taking them out of the data sets and reclustering those. And that's what's shown here, along with all the gene expression analysis. So these populations here are macrophage populations you see in homeostasis. And these on the left here are those you see in disease. You see, they're really mutually exclusive. And in blue here are expression of classic macrophage markers. Excuse me. So we have expression of M1 type macrophage markers in NOS2 and MET. And we also have expression within the same cells of M2 macrophage markers, such as ARCH2. So these macrophage populations that appear specifically in disease appear to be a hybrid type, somewhere between M1 and M2, that you don't see in homeostasis. And we were able to show that in disease, so in homeostasis, you don't really see MET expression in these macrophages. Now, MET, of course, I haven't mentioned, is the HGF receptor, which is why it's of interest here. But the number of MET expressing macrophages, this M1, M2 hybrid, dramatically increases in periodontitis. And those cells expressing MET, those macrophages, as you see here in magenta, are very closely associated with the CD34 positive telocytes. They're clearly the telocytes are making connections directly to these macrophages.
Hold 2 minutes, please.
Okay, this shows an analysis here, velocity analysis. This is the population, and I won't go into that. So does this mean anything? Well, you can inhibit MET very easily using small molecule kinase inhibitor, tivantinib, and ask the question, if you inhibit this accumulation of METs and break down this pathway, what happens? And basically, what happens is, as shown in these bottom slides, that you dramatically reduce the number of ARG1 positive cells, these macrophages here. So this M2, M1 hybrid macrophages are lost if you break this down. So what that means in terms of a model in the PDL is that you have resting telocytes. When you induce disease by whatever mechanism, and we don't know what that is, these telocytes increasing number, they express HGF. At the same time, you produce a population of macrophages that express MET and are a hybrid M1, M2, and this is just after a few hours. You have that interaction between HGF and MET, and that drives this further transition from M1 to M2, so a pro to an anti-inflammatory macrophage. Finally, very quickly in the last few seconds, just to mention Anna's work, we're doing, she's now heavily involved in doing spatial transcriptomics on, this is now on human sections of the gingiva. And she's going to be talking about this at the up and coming IADR meeting, but it seems to be working really well, and we've started to refine it even more using various methods, which I'm happy to mention, just to identify different disparate regions of the tissue itself. This is the tissue here. Thanks very much for, whoops, my slides aren't advancing, listening. As mentioned before, we are an old center. We've been in existence now for 27 years. These are some of the people at our 25th anniversary. The people in my lab who've done the work, Valjani is our bioinformatics guru. Anna, I mentioned, Guy's done the telocytes in the incisor, and Jing has done all the work on the telocyte macrophage interface. Hope that's on time. Thank you.
Thank you so much, Paul. So we do have several questions and Q&A. Please do continue submitting yours, and we will try to address at least a couple live. And Paul, if you have a chance, go and type your answers later. So one of the questions is from Victor Ubels. For the telocyte macrophage connection, as shown using cell chat, was this done using only the murine database, or was this connection also verified in their human database? If so, was this complementary, or were there interesting deviations between mouse and human?
So we haven't looked, we haven't really addressed that question. We've only looked in the data sets we have from the gingiva, where we see something different, basically. It's not exactly the same. We haven't interrogated any human data sets from periodontium. We have our own that we are generating, but we are not there yet to interrogate those ourselves. Mm-hmm. So I don't know the answer, basically.
Thanks. The next question is from Philippa Holly. Do the telesites express connexin 43 or other gap junctions for mechanosensing?
Oh, yes, they do. Mechanosensing is one of the key features that people believe they're involved in. It's probably why they are interacting with the odontoblasts, which are mechanosensory cells within the tooth. Whether they express those specific molecules, to be honest, I can't remember without digging into the data sets, but they certainly express, I think they certainly express piezo molecules. Because we've picked up those, but I'm sure they express equivalents of those, if not those exact molecules.
Great, thanks. And one question from Jamal Al-Kabani. Are the stem cells responsible for oral cancers?
It depends what view you take in regards to oral cancer stem cells. Yeah, I don't think I've seen any clear evidence of that appearing. Obviously, you know, it's not easy to induce oral cancer in mice, I believe, at least not consistently at any rate, which is what you'd really be wanting to do. And I've not seen any data, but in general, if you believe that there is a stem cell basis for many cancers, there's certainly stem cells there. They're in small numbers, I have to say, in many of the oral tissues, certainly in teeth. But I don't know is the answer. I wouldn't be surprised.
Thanks. And one last question from Kai. Is CellSer a functional marker? Meaning, do knockout mice have a phenotype?
So we have a CellSer1 knockout. And just before lockdown, we got the first evidence that suggests it does have a function. Because if we knock it out, we don't see the same induction of the quiescent cells that we did. But the caveat to that is these were just the first round of experiments that we were able to do before lockdown. And then we are now waiting for the mice to start breeding again to answer that question. So again, I think so.
Great. Thank you so much, Paul. It was a pleasure to have
Thank
you. And we're ready to move to the second part of our seminar. We're happy to welcome Magnus Lynch. So Magnus Lynch graduated in medicine from University of Cambridge and obtained his Doctor of Philosophy from the University of Oxford, where he studied epigenetics with a focus on the sequence elements governing recruitment of mammalian polycomb group proteins. He subsequently undertook postdoctoral work with Professor Fiona Watt in the Center for Stem Cells and Regenerative Medicine in King's College London, employing ultra-deep sequencing and single-cell transcriptional profiling in combination with computational modeling to dissect cellular and lineage relationship in human skin. He is currently a research group leader at the Center for Stem Cells and Regenerative Medicine, as well as a consultant dermatologist at Guy's and St. Thomas NHS Foundation Trust. Dr. Lynch's lab is currently focused on combining single-cell genomics with spatial genomics approaches to study just hold on. To study fibroblasts and on developing cell therapy approaches to the treatment of abnormal wound healing and scarring. The lab additionally has an interest in the application of artificial intelligence methods within clinical dermatology. Dr. Lynch, welcome.
Thanks so much for the introduction, Ellen. I hope you can see my slides and hear me. Let me know if you can't. So as you said, I'm a consultant dermatologist, and I'm also a group leader in the Centre for Stem Cells and Regenerative Medicine at King's College London. So I'm going to talk about the work that my group has contributed to within the Human Cell Atlas initiative. So first question is, what do we mean by an atlas? So the dictionary definition of an atlas is a book of maps. And this is essentially what we're aiming to create with the Human Cell Atlas, maps to help us understand the cell types that are present in the human body across multiple organ systems in both health and disease. And this has really been facilitated by the recent technological developments in both high throughput single cell profiling, but also in spatial transcriptomics. So my group has been contributing to the skin aspect of the Cell Atlas. Our work in this area has been largely funded by the Wellcome Trust and has been performed in collaboration with Professor Heminifa's group in Newcastle and Professor Fiona Watt. So I'm sure many of us are very familiar with what a fascinating and important organ the human skin is, so I'm not going to labour the point, but suffice it to say that the skin is a complex organ that comprises in excess of 20 cell types arranged in a complex three-dimensional arrangement with important roles in defence against infection, thermoregulation, fluid homeostasis, and social signalling. This is quite a short talk, so I'm only going to have time to give a flavour of some of the work that's going on, but I'd like to focus on three core questions. Firstly, why do we need a skin atlas and cell atlas in general? Secondly, how far have we progressed towards this goal? And finally, what work is ongoing and what remains to be achieved? So, well, firstly, to answer my first question, why do we need a skin atlas or cell atlas? I think it goes without saying that a comprehensive understanding of all of the cell types in the human body and in the skin and their interactions is of immense academic interest. However, I think the longer term impact will likely be in better treatments for patients and understandings of disease. So my day job, I'm a consultant dermatologist and I see patients with all forms of skin disease. It turns out that actually there's a very large number of different skin diseases in excess of 2,000. And while some of these are common, like eczema and psoriasis, others are actually very rare and as a consequence, difficult to study and poorly understood. And this is an example of a very rare but very serious skin disease called toxic epidermal necrolysis. It's a very rare condition, usually triggered by exposure to certain drugs. It's very idiosyncratic, difficult to predict who will be affected. It has a mortality rate of 30 to 60%. And because it's so rare, even a major centre, such as the hospital in which I work, will generally see no more than a handful of cases in a year. And as a consequence, we really lack a clear understanding of the underlying mechanisms, and we lack an evidence-based approach to treatment. So one of the things which a comprehensive single cell atlas of skin might help us to achieve is to understand rare diseases, both in terms of pathogenesis, but also in targeted and tailored therapeutics. So where are we with the Skin Cell Atlas? So I'm just going to talk through some of the work which has been led by Prof. Heminifa's group in Newcastle and was recently published in Science. So in this paper, in excess of 500,000 cells were profiled across the adult skin, developing skin, and in common skin diseases, eczema and psoriasis. This is a UMAP plot with which most of us will be familiar. Each dot represents the transcriptional profile of a single cell. And of course, cells with similar transcriptional profiles are located close to one another. And these cells can broadly be divided into lymphoid and mast cells, antigen-presenting cells, dermal non-immune cells, and epidermal cells. And if we drill down into these subpopulations, so for example, here is the populations of innate and adaptive lymphoid cells. You can see that these have been plotted separately on a UMAP and clustered separately. You can see that approximately 4 clusters of each are apparent, and these do correspond broadly to recognized immune cell subtypes. Similarly, in the dermis, when we look at the non-immune subpopulations, we can see many of the cell types that we expect. Interestingly, on the basis of transcriptional profile, we can see at least 3 subpopulations of dermal fibroblasts, This is of particular interest to my group, as we have a long-standing interest in fibroblast subpopulations, and a number of years ago we were able to demonstrate that in fact there are multiple functionally distinct subpopulations of fibroblasts within the dermis. When we look at the keratinocytes, which form the barrier layer of the epidermis, the clusters are not as clearly defined. This likely represents a continuum of differentiating cell types rather than distinct cellular identities. We can clearly see that there's a differentiation between differentiated and undifferentiated keratinocytes, and these can be clearly visualized. However, there's potentially some other cell populations present as well, but these do require some additional functional validation. So I mentioned previously that one of the reasons to have a cell atlas was to help us understand skin disease. And in addition to normal skin, Reynolds et al. Did look at single cell profiles in both in common skin diseases, eczema and psoriasis. I don't have time to go through all of the results, but one very interesting finding is that when they looked at the immune populations present in the skin and compared to healthy skin, they could show that there were clear differences when you looked at the sites of the rash. But really interestingly, when you looked at the normal skin from these patients with the disease, even when it was a control skin, a lot of these immune populations were also different. And this does suggest that although the rash may come up in one area, the actual immune profile and the sort of resident immune differences within the skin are across the entire skin surface area. So moving on, working as a dermatologist throughout the COVID-19 pandemic, I've seen quite a number of patients with skin manifestations of COVID-19. And one of the most distinctive is this phenomenon known as COVID toes. It's essentially chilblains that are caused directly or indirectly by COVID-19 infection. Chilblains are essentially caused by inflammation of the small blood vessels within the skin. And a question the question then is whether the inflammation is the consequence of direct infection by the COVID-19 virus, or whether it represents simply an indirect, for example, immune complex mediated small vessel vasculitis. We were able to take advantage of the Atlas data, which is out there for the human skin. So we looked at the expression profile of the ACE2 receptor within all of the different skin cells, cell populations within the skin, and also performed RNA-scope to look at this question. And what we found was that the ACE2 receptor was present predominantly in the differentiated keratinocytes, but also in blood vessels. And this is of interest because it could potentially support a direct response to viral infection in the etiology of rashes in COVID patients. And interesting might also be relevant to acquisition of COVID-19 infection, potentially via a cutaneous route of transmission. So what's next in terms of the Skin Atlas work? So I think the next thing really is we've got a pretty comprehensive catalogue of cell types, but what we need to understand is where these cell types are within the tissue and how they relate to one another spatially. So one way that we can do that is to use methods such as RNA-scope. So here I've shown the different subpopulations of vascular endothelial cells in 3 different publicly available skin data sets, along with markers which are conserved across all of these 3 data sets. And using these markers in RNA-scope probes, we can look at the location of these populations within the tissue. And RNA-scope is a method that allows us to interrogate the spatial expression patterns of genes of interest in intact tissue sections. An advantage over antibody staining is that the detection is highly specific and also very sensitive. So here we can see expression of these vascular endothelial cells in both the papillary dermis and the deeper dermis, corresponding to the 2 cutaneous blood vessel plexuses. We can do a similar approach for fibroblast populations in the skin. And as I've mentioned, my lab has a particular interest in these fibroblast populations. So taking the markers from the single cell sequencing transcriptional data, we can then start to look in the skin. This is a RNA-scope with decarin, which is an mRNA encoded in pretty much all fibroblasts within the skin. It's a good marker, pan-fibroblast marker, and it shows us where the fibroblasts are located. so you can see these are the red dots. And then we can also stain for the markers which I've shown you to try and identify the subpopulations of fibroblasts. And this is work that's ongoing. Now, the thing I would say about RNAscope is it's limited in that you can only look at a small number of probes at once, and actually making sense of that data from the small number of probes is quite challenging because you don't have all of the other cell types identified. So the next step really is more high throughput methods of spatial genomics. This is spatial transcriptomics. This is the 10x Visium approach, and this would potentially will allow us to study the expression of large numbers of genes in parallel. It essentially divides tissue sections into tiny squares and quantifies expression of a large number of genes in each of these tissue domains. A limitation is that it doesn't achieve cellular resolution, but you can infer cellular populations bioinformatically. And to date, we've prepared 10 Visium libraries, which we're just awaiting the results of sequencing. This is an example from the Visium website of what it looked like for the mouse olfactory bulb. So hopefully we'll get similar quality data for our skin samples. Another, as I said to you, a limitation of Visium is that it can't achieve cellular resolution, but other emerging spatial transcriptomic approaches do have this potential. And this is 10x Cartana. This is a targeted approach that uses padlock probes in rolling circle in situ amplification to study a panel of up to 200 genes of interest. And we've been working with 10x Genomics via an early access program to perform this in human skin samples. So this should really allow us to identify at cellular resolution large proportion of the cells that are present within the tissue sections. Okay, so far in the talk, I've made the sort of implicit assumption that in fact the skin is a homogeneous organ, but it is far from the truth. So the dermis, in fact, is in different parts of the body has very different embryological origins. The abdominal dermis develops from the lateral plate mesoderm, the dorsal dermis from the coraxial mesoderm, and the facial dermis from neural crest. And these different origins correlate with dramatically different morphology in different parts of in skin derived from different parts of the body. In particular, differences can be seen between skin of the head and neck, which is derived from the neural crest, and the skin of the body. And the skin of the face and head and neck generally has a much greater content of skin appendages, such as hair follicles and sebaceous glands. This is particularly apparent with this keratin 14 stain in white, where we can see in the leftmost panels the greater content of sebaceous glands and hair follicles, and in the rightmost panel the skin from the arm, where there's very few present. So this is consistently the case when we quantify across multiple donors. So it's a very consistent finding. And we wanted to understand at the cellular level the determinants of this. And in order to do this, we've performed single cell sequencing from 10 skin samples derived from the face. And the analysis of this is ongoing, but essentially in the left panel, you can see the results of this single cell sequencing plotted on a UMAP plot for all of our 10 facial samples. And then in these other two panels, we can compare with publicly available data from the body. And the interesting observation really is that there's not new clusters present within the face. So these are the same cell populations, but they're arranged differently. And the question is how this arrangement comes to be. And of course, we're performing a great deal more work in this area. I can show you to begin with that these cell types do retain a transcriptional memory of their embryological identity, with HOX gene expression specifically certain HOX genes specifically expressed in the body, and the preferential expression of neural crest markers such as SNE1 in the facial samples.
Magnus, 2 minutes, please.
Thanks. So finally, having talked a lot about the molecular biology, our goal is ultimately to understand not only cell identity in vitro, but also to relate this to structure and function in the patient. And towards this goal, we've been using optical coherence tomography, which is a non-invasive live skin imaging methodology that can generate three-dimensional images of both the human skin and blood vessels in live patients. And using three-dimensional reconstruction of this data, we can compare the morphology across body sites, and we have identified very clear differences in morphology of particularly blood vessels across different body sites, and that's quantified here. And in ongoing work, we're relating this to the presence of the different vascular cell subpopulations. So I'd like to thank all the members of my group, particularly Clarice Garnier and Nasrat Harani, who've performed most of the work that I have shown you. I'd also like to thank my collaborators in the Skin Atlas, Professor Fiona Watt and Professor Maz Hanifa, and members of both of their labs. Thanks.
Thank you so much. We do have a couple questions. So one is from Bethany Hughes. Are all your skin donors age controlled, or have you looked into the impact of age on structural skin changes?
They're not age controlled because it's hard to get healthy people to give you skin generally. So as a general principle, we take what skin we can obtain, and there's a range of ages. We are looking at the consequences of skin aging and UV exposure. This is one of the questions that we're interested in. So we are deliberately trying to collect old samples and young samples, but the data I've shown you so far is not age controlled.
Thank you. The next question is from Bruce Arnold. Isn't the lividus reticularis COVID pattern usually seen in the post-COVID period? Is it usually well treated by steroids? And what does that tell us about the mechanisms?
So it is usually seen in the post-COVID period, and without a doubt, it's some form of immunological reaction. I think the question, the question in terms of mechanism is whether it's triggered by direct COVID-19 uptake into the endothelial cells and endothelitis, which then triggers an ongoing immunological response, or whether it's simply a systemic immunological response which triggers an immune complex vasculitis. We don't know the answer to that at the moment. In terms of treatment, yes, steroids is one treatment. Generally, the first line treatment would be topical steroids. Then you would use either oral steroids or aspirin or nifedipine, which is a calcium channel blocker. So I think that the response to immunosuppressant things such as steroids would support the immunological mechanism of the process.
Thank you. The next question is from Hao Yan. Your comparison between cells from different locations looks super nice. Which integration did you use?
I have to ask my postdoc Clarice who did the work. I don't know off the top of my head. It was done on SERRAT, so it was basically done with the standard integration. I think the find anchors is the function that's used on SERRAT, but as to what parameters, I'd have to double check with her. She did all the complex bioinformatics stuff.
Thank you. And a question from Jamal Akabani. An important question, as the skin is the main target in radiotherapy, how skin differs between pediatric and adult patients?
That's a great question. I haven't looked at that, and I don't know specifically. My guess would be that as you age, you lose certain cell populations, and that the tissue reservoir is less. So I would predict that you would lose some of your innate stem cell reservoir, but I haven't specifically looked at that.
Thank you very much, Magnus. It's been a pleasure having you. And with this, we would like to go over some breakout housekeeping rules. Christine, please take it away.